So I will talk today about tags and make a little survey and also underline that with a couple of practical experiences in this context. Before I do that, a very quick introduction of our company. We have been founded in 2001, have some 6,000-7,000 different products available in our web shop, which we are manufacturing our own laboratories and by some 15 selected partners around the globe. We rather focus on high quality rather than on cheap. That's why our complaint rate is very low. We are located in Bavaria, in rural areas, in a town called Marktredwitz, and we understand ourselves as a qualified and competent consultant, accompanying our clients from early research to bulk productions. And our portfolio consists of everything from making peptides, PEGs, and other next generation polymers for drug delivery. And in the last some 10 years, we have collected a decent experience in the field of linkerology. Our slogan is empowering peptide innovation, and particularly for this specific topic of Tag-Assisted Peptide Synthesis, I will pick a couple of examples and explain what we mean with that. So we are focusing on some building blocks for making peptides, and that will be the topic of my talk of today. So empowering peptide innovations means that as we are attending lots of conferences, we see lots of innovations from different people, mostly academic persons. And then if we see that is useful for the whole community, then we pick that up, talk to these people, and make it available then for everyone. So now this is the point here. We have a nice peptide, a beta amyloid of pharmacological interest, but you see that the HPLC is basically a disaster. You cannot purify it from its impurities, and this specific peptide has been made on a tender gel resin, so we have a little bit of PEG leaking, which is also over this peak. And the point is basically what to do in this context. Should we just discard it, go to the next project? But there's a possibility to purify such peptides. Will heating help? Well, a little bit, but not really. The peak is now better separated from the PEGs, but still under the main peak there are lots of impurities, and in this manner it does not help. So the solution, or one solution which we found out, is adding, in order to get such a nice sharp peak, is adding basically a tag on the C-terminus, and then this former aggregating peptide will result in a sharp peak and can nicely be isolated and purified. So the question is, of course, which tag to use, which linker, how to put on this tag, and basically also where should this tag then be attached along the peptide sequence. And if you look in general at a peptide, then we have the helping hand technology, which is utilizing amino functionalities, so at the N-terminus or any available lysine within the sequence. And that is mostly been used. That's why I will not go further into details on all the other functionalities. I even dropped histidine and tryptophan on this slide. But I will give much more detailed and practical insights in putting a solubility tag on the C-terminus of this peptide. I also will then briefly mention hydrophobic tag, which we already heard yesterday from other companies and with certain applications. So that's the focus, which I will bring. Helping hand, I will only make a brief theoretical breakup, then practical details on the C-terminal tags and cyclover. So here's the helping hand technology for everyone who does not know it. It starts with normal peptide synthesis, and you will end up basically with some peptide and plus some impurities. And impurities, ideally, you cap with acetic anhydride. And at the end of the synthesis, you can add this helping hand linker, which is in this stage still FMOC protected. after removal of this FMOC, you can add certain lysine, arginine, or PEG linker in order to turn the solubility different than what it is before. After cleavage from the resin, you have your impurities without tag, and you have the target PEP with tag, and this is a decent difference, so that you can easily purify it in HPLC. And the nice thing for in-process control is the removal of the tag happens with hydrazine, producing a chromophore, so that gives you a good possibility for in-process control of this removal of this tag. And you can add it to the N-terminus, or in case the N-terminus is engaged with something else, you can put it on some lysine in case it is available in the sequence. And there are different types of linkers, helping hand technologies available, either with alkyl chain, PEG chain, or even DMB instead of DDE, which will make the tag even more polar and more soluble. And the last one on the right side gives you an example where you can just click on your tag in case you don't want to carry out certain peptide chemistry. So now we're coming to the practical experiences and details. The concept here is we're using a resin with a normal linker, in this case a Siebel linker, then adding a tag. At the end of this tag, we are putting a safety catch linker on it, and then we start the synthesis of our real target peptide. So after the synthesis, we cleave from the resin, the peptide plus tag, purify the tag from all its impurities, and then at the final stage, we are removing the tag from the target peptide. And then we should have a pure peptide, which otherwise would not be able to achieve. So the first question is which tag to use. And if you look in literature, then Steve Kent already has made a couple of trials in this aspect, and he found out arginine is better than lysine, probably understandable with the guanidino group. So in our specific case, we use an hexa-arginine as a tag. And then the point is how to do the connection from the tag to the target peptide. And here we were selecting 3 different safety catch linker. The first is the Noki linker, ETB linker, and you see on the left side how the structure is. Activation of this linker is being done by oxidation, by metachlorobenzodic acid, yielding and then removal by some amines, diethylamine or piperidine. We use piperidine in our case, and that will release the peptide as a peptide acid. So as oxidation is being used in this specific case, we were exchanging the methionine in the middle by an isostructural norleucine, as otherwise it would be oxidized. The second linker we were trying was the Dawson linker. You see the structure on the left side in the second line. Acylation then to this cyclic urea will then turn it into the activated form until it finally hydrolyzes. So here nothing more is happening. You can leave the methionine as it is, and that will yield the target sequence. And the last safety catch linker we were trying out is the Skal linker. Notably, it will give you a peptide amide and not a peptide acid, but we wanted to show this also in this specific context. Activation of the Skal linker has been done by reduction, reducing the sulfoxide to the thioether. And so in this specific case, we were exchanging the methionine by methionine oxide because we were afraid that the normal methionine would eventually be responsible for some premature cleavage. So as we use reduction at the final step, anyhow, we were using methionine oxide, which will be reduced to methionine in the final step, and then we get the target peptide, the peptide which we want. So here is now a large table, and which is summarizing all our experiences. So let me guide you through that because there are a couple of quite a lot of details here. So let's first have a look at the Noki linker. We started, as I said, with the Siebel resin. Coupling of the arginine tag was done with standard methodologies without any issues. Linker coupling, we had a small issue in the fact that we need to do some double coupling until we achieved completion. And similarly, in the coupling of the first amino acid, so alanine on the C terminus, we need to do triple coupling in order to get full conversion. Then synthesis was basically carried out of the target sequence as planned. We saw only a little issue coupling with the second peptide forming decatopiperazine. So we reduced a little bit the time of FMOC removal on this specific case, but on the other positions, it ran through smoothly. So then cleavage from the resin happened without any issues, of course. Purification of the peptide plus tag went very well. We just saw a little bit of sticking to the column, so that's why the smiley is not 100% green, but basically worked quite well. And the final cleavage worked also quite well. We were just hoping that this aggregating peptide is not soluble in water, so that we could simply wash away the tag with water. But surprisingly, this peptide is quite well soluble in water. So that's what was firstly disappointing us, but we then saw that purification by HPLC worked very nicely. If you look at the dorsal linker, we were a bit disappointed. We saw on one side that the activation is not working 100% well, so very well for academic purposes, but if you think about larger scale production, it is not suitable actually, at least with the methods which we have tried. And we later on also saw some linker fragmentation, and in the end, it also did not hydrolyze to completion, so it rather was decomposing than hydrolyzing. So that's why in our specific cases with the dorsal linker, we did not make so good experiences. But the scale linker actually went through very well from the beginning to the end. Only at the final stage, cleaving the skull linker, we then saw that we had the typical fragmentation as we know it from all other benzidryl systems like ring linker. We saw some kind of fragmentation, kind of back alkylation. I will show you the details then in the next couple of slides. So here are the related HPLC profiles. The node-key linker, you see on the left side the crude peptide. We see a little bit of the PEGs from the temperature resin and the impurities. And after HPLC purification, you see on the right side a nice profile with more than 95% purity. Here are the results of the dorsal linker. We see premature cleavage of the dorsal linker and some kind of fragmentation, which is reducing the yield. Purifying works well, so we got some nice results of the purified peptide, but yield was going down drastically. And here's what I mentioned with activating the dorsal linker with the paranitrophenol chloroformiate. It is producing paranitrophenol, which is yellow, but after even 7 washes, we still had some color coming out. So that is has not worked actually according to our expectations in this specific context. Last but not least, the scar linker, as I already mentioned, worked very well. We have a nice peak, which can be nicely purified to over 95% purity. So this actually worked very well. And now we're coming to the most important part, finally removal of the target peptide and the tag and getting the target peptide in pure quality. So here you see the node-key linker, and we actually see that it is separating very nicely. On high retention times, you see the peak of the target peptide. Of course, it looks terrible, but it is pure, and it is due to the fact that it's an aggregating peptide that is the reason why it shows this HPLC profile. But with these methodologies, all the other impurities the tag and linker tag fragment are nicely away from the main peak, and by this manner, this peptide can be purified in a good way. Situation was different already, as mentioned, with the Dawson linker. We tried different pHs, and basically we had some premature cleavage, but on the other hand, the final cleavage did not really happen as we thought, and the product was finally decomposing at a certain time. So with the methods which we tried, the Dawson linker unfortunately did not work very well in this specific setup. And last but not least, the SCAR linker. You see here the structure and activation by reduction from the sulfone to the thioether. Then this benzoyl cation becomes very sensitive to acids and is releasing the peptide amide. We just see, which is indicated with number 3, this kind of C-terminus alkylated impurity. And if you look on the HPLC profile, it is so the target peptide and this specific impurity are actually overlapping. So you might think, okay, is not working, but you can influence that by adding, for example, 1,3-dimethoxybenzene. Then we have, looks a little bit similar, but basically the two peaks are then purely only one or the other peptide. And we have in peak number 1, the pure target peptide, and peak number 3, this specific impurity. And through that, we can separate that and isolate them very nicely. Okay, so just as additional information, with the SCAR linker, you see the structure, how to make peptide amides. But the other example on the right side, with the same concept, the sulfone reducing concept, you see the related linker, which is producing peptide acids. Here we do not have this benzoyl system, so we should also not have this impurity formation. We don't have practical data on that yet, but we'll work on that. Maybe there are a few other impurities, but we don't expect at least so many. So these are the linkers which are basically available. Noki linker, Dawson linker, and the 2 different SCAR linkers in case you want to try it out. So with this, I'm closing the chapter of hydrophilic tags and switching polarity to lipophilic tags, which we heard also already yesterday afternoon. And I'm happy that there was already so many experience be shared with us. And because in many cases, if you're looking on tags like that, the question is, how long can the peptide be made? And yeah, we heard yesterday from the Corton team that actually quite long peptides are also be able to make on that tag. Chitsubo, who invented the molecular hiving, has published icatibant with a 10-mer. And question always was from the audience, how long can the peptide be done? So there are also other tags already in the market from Ajinomoto. Naturally, you have to go to these companies if you want your peptide be synthesized with this technology. We are having the cyclover, which has 4 alkyl chains, so it's supposed to be more hydrophobic. And we are putting any types of linker on it, Wang, ring, whatever you want to do. It is patented and available for everyone, but the good news for the commercial companies is there are no royalties to pay for this specific use. So if you want to try it out in your specific lab, you are free to try to use that. And how does it basically work? If you look on the left side, you see some solution of cyclover with some peptide in THF, and then slowly methanol is being added, and then you will see that slowly the solution becomes cloudy and finally is precipitating. And then, of course, it depends then on the morphology of your precipitate. Is it really crystalline, or is it still gel-like? then you can apply different process methodologies which are being applied as a standard technology also in large-scale applications like liquid-liquid extraction. If it is crystalline, which certainly will depend on the peptide, then you also can think about precipitation and filtration. So last but not least, the green aspect of this technology you see on the bottom table. If we are adding to this conventional solvents one more methyl group, then they turn to become green. So THF to 2-methyl THF is now a green solvent. Toluene with one more methyl group, the methacylene, is a green solvent. And even if you're adding to methanol 1-carbon, it becomes ethanol, again green. Just for your information, and we tested a couple of other supposed to be green solvents in terms of their capabilities, whether they can be used for either dissolving or precipitation of this specific hydrophobic tag, and you see that in the table. The material is commercially available from us, and everyone can use it without royalties, and that is the news which I want to convey to this audience. And with this, I want to close and thank you for your attention. I hope I could give you a little survey about using tags for tag-assisted peptide synthesis, which is demonstrating our philosophy of empowering peptide innovation. Thank you, and I'm happy to take any questions.
Thank you so much, Thomas. It was really, really nice to see the evolution of tag chemistry. May I start with that one question? So the principle of this idea that you finish the SPPS and then you come with a tag attaching to the desired compound and then proceed with solution phase chemistry is assuming that the only thing that you will remove is then capped impurities. The impurities who were capped by the capping reaction. So the truncated sequences. This is the only thing that is not reacting with the tag, right?
If you're using the tag at the end of your synthesis with the helping hand technology. Yeah.
Yes.
Yeah, of course, naturally. The deletion and truncated sequences will still have an N-terminus in case they are not capped.
Exactly. So let's imagine that they are capped, and if you do the job, so there will be, I will say, not critical impurities. They will be quite easily removed by the purification step. So I wonder what is the advantage of having a tag at the N-terminus, because you are not removing the epimers, you are not removing really, really close related impurities. They are coming in the main peak. What is the real advantage of this tag?
No, it helps to remove the truncated and deletion sequences. It does not help to remove the epimers, obviously, yes.
Okay, so we remove the ones which are truncated, the acetylated ones are removing in this way, but we said it's an easy one to remove by the purification process. No, really interesting. I'm really interested to test this one day. I haven't done it myself, but I'm really interested to test.
It gives you naturally more capabilities. If you're using a chiral tag, eventually it has influence on the epimers of your target peptide. So that's why it might be of some advantage to add an oligoamino acid, oligolysine, arginine, or some other chiral functionality. Then you're creating diastereoisomers.
That's very interesting.
I mean, that is more process development, because then you have, there's a reason why the epimer is eluting at the same time as the target peptide.
Yeah.
So, and you need to influence that in that manner that this little difference becomes larger. but that is very sequence specific.
Agree. Any question? Any further question?
On the left side.
Yeah, hello. Yes, it works. So thank you for your presentation. I have a question with regards to the Dawson linker. So essentially, it seems to be working through two different steps. So the first step is the basic condition, just to get the chloroformate reaction, and then you treat with the acidic condition. Do you see that there's one or the other step that's limiting the proper cleavage, or in terms of poor result that he seems to have observed, and which would be the way to potentially improve it in the future?
Well, we were using the conditions which were published by Phil Dawson, and they did not work in this specific case. I easily can imagine that with certain investment in process development, that this will turn out to be a linker which works very well. So I don't want to say at all that the Dawson linker is a bad linker. No, it's just with the methods which we have used, and these were the standard published one, it did not work, so it gave the results as they were. But if you're adding some scavengers, turn polarity of the solvents, concentration, invest in process development, I definitely can imagine that this can eventually be a very good solution.
My question was going in a similar direction. So you showed in the middle of your presentation the treatments you did that gave you this colorimetric readout, that the hydrazine treatment, I believe, where you needed 7 treatments and you still saw a reaction forming. So was this due to really slow reaction kinetics, or is this a mass transfer issue? Because
you mean the activation of the Dawson linker?
Exactly.
I don't know, to be honest. I cannot give a sufficient explanation for that. Okay. It was just the observation which we made. What is the reason for that? We would have looked more into details of that, which we did not do.
Okay, thank
Sorry.
you.
Yeah.
Any further question? If it's not the case, then we go to the next. Thank you, Thomas. Really nice.